My name is Kristina Fite. I am a master's student here at UNT. I do my research on how a toxicant known as 6-PBDQ impacts amphibian larvae. 6-PBDQ is a byproduct of a product known as 6-PBD, which is used in vehicle tires. So it's all over the environment. So it's super important that we know how it's impacting amphibian populations and ecology. So first, I want to just show that we have both 6-PPD and 6-PPDQ. So our 6-PPD is actually the parent compound. It's actually in this little blue bottle that's back here. It is a solid. It's a pellet and it's purple. The nasty little container back there is actually what it looks like when we put our 6-PPD pellets into solution. The 6-PPDQ is actually our tiny red bottle here. So this is what the 6-PPDQ looks like once it goes into solution as well. So when I say solution, I just mean the 6-PPD and the 6-PPDQ are both in solid form. We have to get it into a liquid form, so we actually use a solvent known as dimethyl sulfoxide. And once we use a solvent, that's how we get the 6-PPD and 6-PPDQ into solution. This is the fume hood that I do my exposure dishes inside of. All of my experiments are done inside the fume hood, and that's just because, of course, we're working with a toxicant, both in its physical form and in liquid form, and I just want everybody to be safe when we're working with this toxicant. So those are the 6-PPD and 6-PPDQ. The 6-PPDQ, our tiny little bottles there, we actually put them in the freezer. And so we have a lot going on in here. We have multiple dilutions of our 6-PPDQ. So this is a stock solution that we've created that stays in here, and that's frozen as well. And we also take water samples of each of our exposure dishes. So these little jars back here are all filled with water samples from the test that we have already completed. So that's cool. So one of the key aspects of my project is making sure that I keep all of my exposure dishes clean. So these are dishes that need to go into the muffle furnace. What happens actually first, when a test is being conducted, we have exposure dishes, which are these, inside of the fume hood. So let's say we'll have 6 exposure dishes inside of the fume hood. When one trial is going on, that trial lasts for a total of 24 hours. After that 24 hours, each of the dishes gets emptied. Then it goes through a 3-solvent rinse just to make sure that we have all of the toxicants off of it. Next, they sit out for 24 hours so that they can dry. And once they're dry, they come over here. So these have to go into the muffle furnace. So what happens is we will wrap these dishes, these little dirty dishes, we'll wrap them completely in foil, and then we'll put them inside of the muffle furnace, and this is the muffle furnace. The muffle furnace will then be turned on. It gets as high as 450 C. So what it looks like, those are dishes that have already gone through the muffle furnace, so they're completely wrapped so that they can expand, and if one breaks, the glass is just going to fall into the aluminum foil. But once they go through this process of being in the muffle furnace and they're cooled off, then they come over here and they get unwrapped for the majority of the part, but we keep a small little bit of aluminum foil on the top just so that debris that's falling can't fall into our exposure dishes. So these are the clean dishes that are ready to use for the next trial. Okay, so let's talk about the animals that I'm working with. Each of these bins has amphibian larvae inside of them. So these bins we will clean 3 times per week, Mondays, Wednesdays, and Fridays. And sometimes we'll do a spot clean on Tuesdays and Thursdays. And that's just to make sure that we maintain really good water quality for each of the amphibians that we're working with before they get put inside of the exposure dishes. So these are all bullfrog tadpoles right now. We have a ton of bullfrog tadpoles. These guys are actually a little larger. Their water will get cleaned after this. It's a little dirty, but they'll get cleaned after this for sure. So these are all of our larger tadpoles, and we also have newer, smaller little tadpoles hatchlings, I guess, if you will. And those guys look really, really tiny. So compared to the big ones over there, these guys are really small and they're growing as well. We also have a new type of experiment that I will be starting very soon. And I will be looking at how amphibian larvae change their color in order to camouflage with their background. Post and pre, or I guess pre and post 6-PBDQ exposure. So the research question is, does 6-PBDQ impact amphibian larvae's ability to change their color? So here we have amphibian larvae that are acclimating to white backgrounds, and that's why the bins are painted, and that's why, of course, they're on the white background. And then these guys are acclimating to a black background. So yes, these guys are going to be used in experiment. They're just acclimating right now. Acclimating just meaning they're getting used to their environment that they're in because they're not out in the wild right now. So we need to make sure that they're well rested and they're happy before we actually subject them to the toxicant, I guess.
So why is the research you're doing at the moment important?
The reason my study is important is because the toxicant that I am examining is ubiquitous in the environment. So that just means that it is found throughout the entire environment. So we have seen it in water sources, we've seen it in soil, we've seen it in food, so fish that have been exposed to the 6-PBDQ. And also we are starting to see it in human urine. So it's all around us, it's impacting us. We need to know how it's impacting us.
Wow, that is awesome. I did not know that.
Wow. Yeah.
So what sparked you into choosing the current research that you're doing here at UNT?
I was very interested in toxicants in general. And I wanted to see how toxicants specifically impacted ecology and organisms, of course, in the environment. And luckily, my advisor found out that there was a toxicant known as 6-PBD and 6-PBDQ that was impacting coho salmon. So it was actually him that was like, hey, I know you like to work with toxicants. I know you like ecology. Do you want to work with me on this project? And I was like, yes, that's amazing, actually.
That's awesome.
Yeah, so that's how I got involved. Yeah.
Oh, wow, that is awesome to hear.
I know, I was so lucky.
Yeah. So what does a typical day for you look like, especially when you're doing your research?
Yes. Okay. So there's two very different versions. Yes. There's the lab version, and then there's the outdoor version. I'm going to start with the outdoor version because that's really important. So essentially, we don't just have tadpoles on hand. What normally happens is that we will get a group of people together and we will go out to different ponds around town, sometimes Lila, sometimes Lurf. Lurf is a really good spot, and we will actually look for breeding pairs of adults. So the field work starts at 8 p.m. and we go out in groups. We have headlamps on, we have gear on, we have little bags, little bins for collecting our froggies. So we'll put all of our gear on. Sometimes we have waders, sometimes we have hip waders, sometimes we have full waders. We will go inside the pond. We will look to see if there are any calling males. So the species that I'm specifically looking at right now are the green tree frogs. Very, 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 there's a really great population of green tree frogs at Lurf in Lila. So essentially what we would do, we would go inside of the pond. We will see specifically, and of course here, all of the calling males. And that's how we know where to find the males.
Oh, yeah.
Yes. So then once we find the males, we'll either see one, is he about to be amplex with a female, just meaning that they are together doing what amphibians do. Or we will see if there's a calling male that is attracting a female. So there's a difference between the calling males and the females. So green tree frog males will have a wrinkly neck just because when they're calling, the neck skin gets
does it expand?
Expanded. Yeah, it expands and it basically gets translucent after some time of just calling. Whereas the female green tree frogs don't call at all. So their neck is nowhere near as wrinkly and it's like a really nice cream color.
Oh, wow.
So what we'll see a male calling out there, and we'll usually see a female like creep up to start the amplexing, and we will grab both of them.
Oh wow! Just like that.
Yes, we will grab them. We will put them in bins with a little bit of their water and some substrate, and we will have them in these bins. And we'll wait for them to deposit egg masses. And it's those egg masses that we usually take to the lab here, and we will put the egg masses in a really clean, nice bin. And then after that, the egg mass will hatch, and we will have all of these amazing little amphibian hatchlings everywhere.
That is awesome.
That's what field work looks like. And it usually, again, is like 8 p.m. to maybe 12 a.m. Is usually the time that we're out there. The lab work is usually just prepping, getting started with the exposure dishes, doing calculations, making sure everything is correct, and then putting everything in the fume hood, taking the toxicant out to thaw, and then we get to work on that. So two very different parts of my research, but both are really fun, and that's usually what a day in my life looks like.
Yeah, no, that is awesome.
Yeah, it's a lot.
So how do you feel about the current findings or accomplishment that you guys have had here as you're doing research? If there is any.
It is a good thing. I have found so far that my amphibian larvae are actually not dying at the highest environmentally relevant concentration.
Oh wow. That's awesome.
And we've actually gone a little above the highest environmentally relevant concentration, and they're still not dying from what we know so far of the 6-PB-DQ. It seems like they're actually more impacted by the DMSO once we increase the concentrations.
Oh, okay.
So right now, we know that there is no acute mortality happening for amphibian larvae when they're exposed to environmentally relevant concentrations of the 6-PB-DQ. However, my next step is trying to find, are there cellular processes that are impacted? Because essentially, there's been a paper recently that showed that the 6-PB-DQ may impact the blood-brain barrier of some of the coho salmon. So there might be something underneath that might actually be going on. So that's the next step. But essentially right now, it's good to know that at environmentally relevant concentrations, the amphibian larvae are not having acute mortality.
That is awesome to hear, yeah.
Yes, yeah. So yeah, we got very lucky. Yeah.
What do you hope to find at the end of your research? Is there like a question you want to answer, or is there like a solution you're looking for?
Yes. Is 6-PB-DQ causing acute mortality? Similar to how it has been shown to cause acute mortality in coho salmon was one of the biggest questions that I had. So that question is answered. But now I'm hoping to find out what other processes are impacted by 6-PB-DQ. So one of the things I'm specifically looking at is swim performance. I'm wondering if 6-PB-DQ impacts the swim performance for these amphibian larvae. So that's the next question that I'm hoping to find out by the time I'm finished with this research.
Oh, that is awesome. Yeah, I hope so. I hope you find your answer. Yeah.
Thank you.
How is or will your research create a real world impact? If that makes sense.
Yes. So amphibian larvae are really good for ecological systems. So we know that their larvae period is all aquatic. They are a really good source of food inside of the food web in general. So they are a part of ecology. They are a part of natural ecosystems. The same can be true for their adult stage, which they then consume other insects or stuff like that. So essentially, I'm hoping to get the point across that it's really important that we know what's happening with amphibians, because we want to make sure that we are maintaining really well water quality. We want to make sure that we are maintaining natural ecosystems so that they can continue to function normally. That's what I'm hoping to contribute to the world, I guess, in general. And amphibian conservation, because sadly, amphibians are one of the most threatened vertebrate groups on Earth. So more research needs to be done so that we can continue to see these cool animals.
Yeah, that is awesome. Yeah.
So they stay in this nice, beautiful world of ours.
No, that is awesome. Yeah. So what do you think is the most interesting part of your research?
I think it's interesting to work with a toxicant that is man-made.
Oh yeah.
That is currently, as we know it, maybe impacting the environment. I think that's pretty cool.
That is pretty cool.
Yeah. Because it's just like, we are going to continue making things as humans. We have to make sure that we understand what we're making and what impact it has on the environment.
That is awesome.
So,
Yeah,
yes.
I agree with you. Yeah. What do you use to motivate yourself, especially when there's days where, let's say you don't want to get up from bed and be like, oh, I have to go do my research?
People.
Yeah.
I love working with people.
Yeah.
I love collaborating.
That is awesome. Yeah.
So I have probably like 2 or 3 different side projects that I want to do that I'm collaborating with people who, and their expertise can help me understand certain aspects. So for instance, what I was saying about the swim tunnels, there's another grad student that works with swim tunnels and she knows all about them. So I'm working with her so that I know how to use the swim tunnel so that I can understand how swim performance is, I guess, studied for amphibian larvae. So awesome. I just love working with people, especially people that are passionate about their research.
Yeah.
That's very fun.
That is awesome. Yeah. Okay.
Very motivating.
Yeah. What is your favorite thing that you do when you're not doing your research, like on your free time?
That's a very good question.
If you have any free time.
Yeah, I was like, I don't know if I have very much free time anymore. But I will say that there are 2 things I love. I'm terrible at violin playing, but I love learning how to play the violin.
Oh, that is awesome.
So that's something on my on the side that I'm not doing. And also, I actually have a bearded dragon and a snake. So like, I'm always interacting with them. Yeah. I love buying stuff for them, which is kind of bad, but I love them. I love them so much. I love their environment. That's
awesome. Yeah.
I put a lot of energy towards making sure that they have a really good habitat. So it's become a side hobby for me.
Oh wow, that is awesome.
Yeah.
And you see them every day. They're at home.
Mm-hmm.
That is awesome. Yeah. Okay.
Yes, I'm gonna continue building enclosures for them and learning about them and trying to figure out how I can make them the most happy. That's what I've been obsessed with lately.
Yeah.
But it's been fun.
That's awesome. Fun. Yeah. Okay. Yeah. Those are all the questions. Thank you so much for taking your time to talk with me and to letting everybody know a little more about your research and what you do here at UNT.
Yeah. And if anybody's interested, my advisor is Dr. Jason Bohenek. He's a very great advisor. He works in herpetology. I think he's a good advisor. So contact us, either me or him, if you ever want to get involved in our research. So yeah.
That's awesome. Yeah. Well, you guys heard her. Okay. Well.